Good afternoon. My name is June Axif. I'm the co-founder of MyEye, which stands for My Information. We are developing the next generation of protein detection technology to empower data-driven biomedical discovery. We are in the age of genomics, which has unleashed an unprecedented amount of information about the human body. This has also brought us into the age of omics, where we are no longer looking at single genes, but the entire genome as a whole. However, DNA information alone is not enough for understanding disease. This is because what actually matters is the proteins made from the DNA. We have the same DNA in every single cell in our body, but it is when and where certain proteins are made that inform us of disease and how we age. Indeed, the majority of therapeutics on the market today target proteins. But the reason why my team is interested in learning about proteins is for patients like Tanner. Tanner had a germline p53 mutation, one of the worst mutations in cancer. Germline means that every single cell in his body was a ticking time bomb to become cancerous. And unfortunately, Tanner passed away from brain cancer. And in his passing, his family created this project with the J. Craig Venter Institute and my co-founder, Nicholas Schwark. The project tries to detect cancer at its earliest stages by looking at changes in proteins. This is the type of personalized medicine we want to enable. But to get there, we need to develop the next generation of protein detection technology. So what is the problem? Well, protein detection technology today is cumbersome, extremely expensive, and not sensitive enough. And this is very unlike the recent developments in DNA and sequencing, where today you can sequence all of one's DNA and RNA very cheaply. Our team realized that the fundamental difference is because DNA and RNA can be replicated by PCR technology, polymerase chain reaction. Proteins cannot be amplified directly. There is no PCR technology for proteins. So our team asked, what if there can be? And that's exactly what we have done. So we can take a molecule that binds proteins, such as an antibody, we can put a DNA tag onto it, and now we can amplify this tag using PCR technology to amplify from one strand into millions, thereby allowing us to detect low amounts of proteins much easier. Additionally, because these tags can be unique barcodes, we can now look at a bunch of different proteins in the same reaction, so thereby making this system high throughput and cost effective. So from a single biological sample, we can look at a bunch of different proteins with our cocktail of antibody DNA conjugates. We can collect and then amplify by PCR, and then we can run it on next-generation sequencing to determine the identity of the proteins and its concentration in the sample. And we can leverage existing next-generation sequencing platforms like those of Illumina and Oxford Nanopore. So effectively, we have repurposed all of DNA technology for the use of detecting proteins. And because we're using next-generation sequencing, we can look at DNA, RNA, and proteins at the same time, making collecting all this data very simple. So what is our product? Our first three products are different protein detection panels that look at different types of proteins, and this has a value of $3 billion today. But the true value is in the data that we can generate. So using our panels, we can look at samples from healthy and diseased individuals. We can look at the proteins in the context of DNA and other information, and begin to build this complex relationship map. So this map will finally give us a full picture of a disease and allow us to devise an intervention. This kind of data aggregation is unique to MyEye, and our team has the expertise and passion to solve these kinds of problems. So our business model starts with selling protein panels to academia and industry. We generate data to build our MyEye map using internal and through our partnerships. And then we can license this MyEye map to other institutions for the development of diagnostics and therapeutics. And because this is a positive feedback loop, our customers are constantly helping us improve our products. Our protein panels initially address a $3 billion market, leading into a $24 billion market. And our MyEye map addresses a $77 billion diagnostic and therapeutics market. We have gotten incredible traction and interest through our proof of principle studies, and we are partnering with institutions to develop our panels, such as antibody companies, next-generation sequencing companies, and direct users of our kit. Compared to our closest competitors, we differentiate ourselves by leveraging all the next-generation sequencing instruments already in laboratories around the world. So without having to use specialized equipment, that makes our assays cost-effective and adaptable. Additionally, we are the only platform that can look at DNA, RNA, and protein concurrently in the same sample. We have an amazing team that stem from the Scripps Research Institute and the J. Craig Venter Institute, and we are uniquely able to deliver our technology and build our company. We have accomplished scientists in bioinformatics and personalized medicine, Nicholas Schwark and Christopher Nazer. We have experts in bioconjugation and nucleic acid chemistry, Devin Kayer, Reza Gadiri, and myself. And we are joined by a seasoned business officer, Maria Ferrero. We are looking for $1.5 million in seed funding, of which a third has been committed. Join us in unleashing the next wave of biological data. Thank you.